Good morning. Welcome to video number 16. This is gene regulation in eukaryotes. Epigenetics is the focus. Because we kind of hit transcription factors in that a little bit in video number 14, but we'll come back to that just briefly. The free response that deal with this topic, 2009 number 4 and 2019 number 7. So 2009 number 4, 2019 number 7. Just like before, if you go on our Canvas page and look in files, there's a file that says AP Bio FR referenced in YouTube vids. And so you want 2009 and 2019, number 7. All right. So to start off with, if you do have your old notes with you, I know I don't always remind you of this, but there's a spot that says gene regulation, eukaryotes. All right, so to start off with, if we look inside of a nucleus, then of course genetic material is in there. And we learned previously the DNA will be wrapped around histones. That way it's less likely to get tangled. In addition, when DNA is wrapped around histones, the DNA is more likely to be off. Because the idea is, and we also touched on this briefly in a previous video, the idea is if this is my DNA double helix, then if it's sort of like this, I can unzip it, do transcription, later translation, and the genes are on. I'm building whatever proteins, I'll pick the right words at some point, whatever this genetic code is telling me to build, I'm building. Let's say it's a protein, for example. But if that DNA is coiled around histone proteins, if my hand's the histone, then it's much tougher to get in there and start unzipping things. So the more tightly condensed DNA is around histones, the more likely it is to be off. And we can see that in a real nucleus. I'll zoom in another click or two. Inside this nucleus, the nucleus is here. If we completed the circle, there's the nuclear pore where RNA would leak out after we've done transcription and 5' cap, poly-A tail, splicing. You'll notice inside, some of the DNA, all the DNA is stained black, but places where there's a ton of DNA all cramped together, it looks really dark. Places where it's more loose, it looks further apart. Kind of like at a OU Texas football game or something like that. If I look over here and it's just a sea of red, then it looks red. But if every other seed is full, then now it might look like kind of a lighter red. The more pixels of red I have, the redder something looks to some degree. Not a perfect analogy. So here, the DNA is very, very compact. Those genes are more likely off. So let's say that this gene was, that this nucleus was from a skin cell. Well, are there genes that code for heart and brain and lungs in a skin cell? Sure. So every cell in our body, with a few exceptions, red blood cells eject their nucleus. But every cell in our body should have 23 pairs of chromosomes. I think liver can be quadruploid sometimes. But anyway, so in general, we've got all 23 pairs of chromosomes. So we've got all 21,000 genes in every cell. But the reason a skin cell and a brain cell are different is because in a brain cell, the brain cell genes are on. And in a skin cell, the skin cell genes are on. So in this skin cell, all the code for brain and lungs and whatever is off. These genes right here are tightly condensed, so we're seeing them coiled up around histones, so they're more likely off. And that is called heterochromatin, where it's kind of dense. Euchromatin, where it's less dense, those genes are more likely on. Okay, so now let's discuss histones a little bit. DNA wraps around histones up here. I've got histones down here. I kind of drew a picture. This is a two-dimensional picture. You can see that there's actually like 8 spheres of histone proteins that make up a overall histone. And they number them like histone, like H1, H4, and stuff like that. So my DNA will take 2 wraps, 1, 2, 1, 2, 1, 2, kind of 2 cycles around histone, and then it'll pack down. And then that DNA will then fold, and then that DNA folds, and that DNA folds, until eventually I can get 6 feet of DNA into a microscopic cell. And so here are some microscopic images. I mean, they're not microscopic now, they were. So there's the DNA double helix, and here's what it looks like under a microscope. Then when we start attaching the histones, it looks like beads on a string is how we always refer to it. And then I can start packing those down. So a histone, or the histone proteins and DNA together, is called a nucleosome. That name probably doesn't matter too much on an AP Bio test. But I start scrunching those down, and I pack that down, and I pack that down. And then eventually I get to the more familiar chromosome like we're used to seeing. And this is actually like two sister chromatids joined together. So there you go. That's what a chromosome looks like. But it all starts with DNA wrapping around histones. So why would DNA wrap around histones at all to begin with? Well, you might remember, if we go back and look at the chemical structure of DNA, all along the edges of the DNA double helix, I have phosphate, sugar base, phosphate, sugar base. A nucleotide is phosphate, sugar base, phosphate, deoxyribose sugar, and a nitrogenous base. Well, if I have phosphates, phosphates have negative charges. When a phosphate's on its own, it's got 3 negative charges. When it bonds with something, it's got 2 negative charges. When it bonds with something and something else, it's still got 1 negative charge. So all along here, your DNA, whether it's yours, mine, that plant outside or whatever, we all have negative charges on the outside of our DNA. So if I want DNA to stick to histones, what would I assume about histones? Right, if DNA is negative, then maybe some parts of histones are positive. And so if we start looking at the structure of a histone, it looks something like this. So you'll notice these little tails. These little tails are charged. So right here, in this picture on the left, this is my DNA, 1, 2, and you'll notice those little tails are kind of like reaching over and sort of holding the DNA down. So the DNA is wrapped around histones. This is my double-strand DNA. It's wrapped around histones, but I also have these tail-like structures that are sort of like taping it down. So it's almost like I take this and this pen and I wrap this cord around it and then actually apply some tape to like hold it down. Now it's going to be hard to access that DNA. That DNA is off. The genes are off. Well, essentially what I can do is I can acetylate. So an acetyl group, it's an organic chemistry group. Looks like this, C double bond O, and then CH3, a methyl group. That's an acetyl group. A methyl group is just CH3. We'll come back to methyl groups here in a little bit. But essentially what we'll do is these little positively charged tails, if we add acetyl groups to them, it's like it's covering up the positiveness. So the example that I've given in class several times is sort of like this. This doesn't have tails like this, so this is a less complex version of the histone that you see on the screen. Let's say that this paper towel spool here has a bunch of positive charges. My DNA is negative, so DNA is going to stick to that. Okay, well now if I go in and I get some chemical groups called acetyl groups. So let's say that these are some acetyl groups, C double bond O, CH. Now, if I cover up those positives, it's not positive anymore. So I start covering these up with acetyl groups. Now my DNA is not really attracted, so it's not going to stick. So now my DNA will kind of fall off and become loose. Now if my DNA is loose, hang on, there we go. Now if my DNA is loose, I can unzip it, read it, transcription, and then translate it. So overall, in summary, histones have some positively charged tails. DNA is always negative because of the phosphates. If I add acetyl groups to those positively charged tails, it's covering up the positiveness, so to speak. And so now the tails let go of the DNA. Now the DNA is loosely packed and the DNA is more likely to be on. So if I acetylate histones, then that's turning genes on. If I deacetylate them, if I take the acetyl groups away, then the reverse happens and now I'm turning those genes off. So this is one way that we can manipulate histones. We're just going to talk about acetylation. Turns out you can also methylate, you can also phosphorylate histones, but for the sake of confusion, we're just going to focus on like one thing, acetylation. Okay, so the next thing that we want to look at is methylation. We can also methylate our DNA. And It kind of changes all the time, or not all the time, but I believe the consensus is still acetylating and deacetylating histones is a little bit easier or a little bit more flexible than methylating DNA. So whenever we methylate DNA or demethylate DNA, that's a little bit more permanent. It's not permanent, we'll see, but it's a little bit more permanent than acetylating histones, if that makes sense. Okay, so you might remember from a previous video, video 14, transcription and translation. Whenever we're going to transcribe DNA, RNA polymerase has to land on a promoter, and then it'll start transcribing the DNA. Okay, well, same thing over here. Let's say I got this gene, this red one. I have a promoter, and right now that promoter is, we'll just say, like open. Well, what I can do, so my RNA polymerase could land on there and start reading. Yes, we might need some transcription factors and some activator proteins and stuff like that to help. But technically, my RNA polymerase could land on there and start reading. Well, what we can do is add methyl groups. So when I start adding methyl groups, again, methyl is CH3, CH4 on its own is methane, CH3 is methyl. When I methylate the promoters, then now, if my memory serves me correctly, now proteins go and land on those methyls. And now my RNA polymerase can't. So when I add a methyl group to a promoter, proteins then glob onto it, and now that promoter is basically blocked. Remember, a promoter is a parking place for polymerase. So if we go and we put somebody in your parking place, then polymerase can't land there. So we've effectively turned this gene off. The reason that matters is, again, maybe this is maybe one of these codes for muscle and one codes for bone. And let's say I just injured some muscle. Well, if I took a fastball to the thigh or something like that, and my muscle cells are damaged and I want to regrow new ones, maybe I need to turn on the muscle growing genes. I don't want to turn on the bone growing genes. So I want the bone genes to be off. Okay, there's actually a disease. I think I've got it in here. You might remember we saw this earlier in the year. But remember this guy? We start looking at a bone density scan, and you notice he has bone growing where his muscle's supposed to be in his left thigh. There, we can see it in his left thigh. Let me zoom out a click or two. That's his left knee. His left hip would be right up about here. His waistline would be right about here. So you're seeing right about here, bone is growing where muscle's supposed to be. So he has MOP, myositis ossificans progressiva. Myositis, myo is muscle, itis is inflamed. Ossificans, ossify is bone, and then progressive, it becomes worse. So it's a progressive disease where muscle is inflamed and it actually becomes bone. So let's say that this up here is a muscle cell and it gets damaged. And so now, or in this area, there's a bunch of muscle cells and they get damaged. So this one is still alive and it's going to divide and repair the damage. What it's supposed to do is have all the bone genes off and turn on the muscle growing genes. But what if it does the wrong thing? What if it turns off the muscle genes and turns on the grow bone genes? Then what do you grow? Bones. So this is a mistake. This is where we turned on or off the wrong genes. Okay, so back to this one. I've got my promoter. I can add methyl groups. So if I add acetyl groups to histones, then that takes away the charge, and now the genes are more likely on. If I add methyl groups to promoters, now those genes are off. Okay, well, does it matter that much? First of all, you probably can't see this very well, but promoters tend to have a lot of, we call them islands of G and C, so it'll be like CPG, and for phosphate, cytosine, phosphate, guanine. And so what will happen is we can methylate those C groups. That's beyond AP Bio probably, but there are specific things that we methylate there. So there's a promoter. All the little yellow spots, you probably can't see it very well, are C's and G's. Over here, the yellows, C's followed by G's are less. So these are all opportunities to methylate the promoter and turn it off. Okay, so first off, if you're kind of following along in your notes here, number 1 was methyl patterns are significant. Scratchy throat. No dry cough though, so that's good. Okay, so with Calico Cats, it turns out whenever you look at Calico Cats, you see this weird phenomenon. A male can be orange or black. A female can be orange or black or calico. Calico is where they're a mixture of orange and black. In dogs, it'd be similar if you know what brindle is, and a dog like boxers can be brindled. And so what's happening is this. We'll come back to this picture in just a second. But what we'll notice is if you look at a cell, this goes for a cat or humans. If you look at a mammalian cell, females always have this little glob inside of their nucleus. Males don't. So like this right here, I'm just looking at a cell from a female and I can be like, that is a female. Even if I'm not analyzing like, does it have X chromosomes and Y chromosomes? Like I'm not doing a karyotype or anything. So what is that? Well, it turns out that only one X chromosome is on in a normal cell. Apparently, if they're both on, that's too much protein. Kind of like if I'm supposed to have 2 chromosome 21s, and instead I have 3 trisomy 21, now I make extra protein when it's time to read any genes on chromosome 21, and that ends up with Down syndrome. Well, if you're a male, you have 1 X chromosome. The X chromosome still has genes on it for building like regular stuff, like kidneys, lungs, organs, stuff like that. The Y chromosome just makes female parts turn into male parts, so like reproductive organs. So a male only has 1 X chromosome. A female has 2. But in a female, let's make these different colors. In a female, in each cell, one of those X's is turned off. So these are not identical. Yeah, they're both X's, but they aren't identical. So you might have a capital A for some gene on this one and a lowercase a for some gene on this one. Well, in a female, there's a female nucleus. In a male, the X is on and the Y is on. There you go. In a female, one of those X's is going to be turned off, and it's just turned off at random. So essentially, one of these X chromosomes gets covered in protein and just slammed against the wall of the nucleus, and they call it a bar body. So that gene is totally off. So in that cell, these are the genes that are on. In the cell right next to it, it might have gone the other way. This one got turned off. So they'll be like heavily methylated and then covered with protein and then like shoved against the wall of the nucleus. So that is the covered up X chromosome that you're seeing right there. You don't see it in males because we don't shut off one of our X chromosomes. So what if this codes for brown fur or dark fur and this codes for orange fur? This cell will produce fur that's orange. All the fur coming from this one will be brown. And that's what you're seeing in this picture. If you're a male and your X chromosome contains the color capital B, codes for orange apparently, lowercase b codes for black, okay. If you're a female and you get a gene for black and black, you shut off one of them, black fur. Orange and orange, shut off one of them, orange fur. Zoom in just a little bit. But then if you go over here, if I've got an orange one and a black one in this cell, the black one is on, and this one, the orange one is on. And so the final result is that little patch, they all came from these cells. So this cell turned into 2, 4, 8, 16, 32. This one turned into 2, 4, 8, 16, 32 to become the rest of their fur. So the whole point of this is methyl patterns matter. And not just like in some slight sense, like methyl patterns can change the phenotype. So like that cell right there that's producing black hair is genetically identical to this one that's producing orange hair. It's just which genes are on and which ones are off. So epigenetics is above genetics or covering genetics. So genetics is DNA, A's, T's, G's, and C's, RNA, A, U, G, and C. Epigenetics is, are those genes on or off? And so what the whole point of this is epigenetics can still change your phenotype. Phenotype is what natural selection acts on. So yes, With natural selection, it's only the genes that I pass on to my kids, but nature is selecting based off of phenotype. So for example, pretend that brown eyes gave you some sort of mating advantage. I'm not saying looks, it just, we don't, I don't know, brown fur or something like that. But brown eyes, pretend that it did. Okay, so if you're big B, you got an advantage because you have brown eyes. Pretend there's only brown and blue, and brown is dominant. Pretend that that's how simple eye color is. If I'm big B, little b, I've still got brown eyes, so I'm still more likely to survive and pass on my genes. So yes, I pass on my genotype, but nature's selecting my phenotype. So whenever epigenetics changes your phenotype, that matters. So first off, methyl patterns matter. Next up, the second one is methyl patterns switch throughout life due to experiences. So over here we have mice, and baby mice are called pups, kind of like dogs are called pups. There we go. And it turns out if you got this pup, if it's raised by a mother that's anxious, then it becomes an anxious adult. Okay. If the pup is raised by a nurturing mom, she licks him, grooms him, cuddles with him, then that mouse becomes a relaxed adult. So scientists were trying to figure out, okay, why is this? In psychology, it's always nature versus nurture. So here's a study if you were kind of interested. If you just Googled Science Daily and you Googled that title, Maternal Care Influences Brain Chemistry into Adulthood, Animal Studies Show. This is from December 2011. Down at the bottom of the study, they'll have a link to the real study. So that would be the actual scientific journal article. And what they found in this study was this. Okay, we've got mom. And what they did was they went in and they found some genes that they think are the cause of anxiety in mice. And it also causes general stress. And so there are other problems that come with that too, like releasing cortisol causes stress, and stress can lead to heart attacks. So for example, in humans, I don't know about mice. So they went in and they engineered some mice. They were all genetically identical. And they put methyl tags on there. So they go in and this particular gene, NR3C1, we're not worried about all the names, but that particular gene right there, we methylated the promoter. So that is, has 2 methyl groups on it. And it turns out, when they're methylated, that those kids, if mom doesn't lick them, if mom doesn't groom them very much, those methyl groups stay. And those kids end up with high corticosterone levels, high anxiety, and they go on to kind of ignore their kids. They don't lick them, they don't groom them, they're not very cuddly. And so their kids will have the methyl groups, and the next generation will have methyl groups, and the next generation will have methyl groups. Okay, then what they did was they took the same types of pups, identical pups with the methyl groups, they put them with a mom that is very attentive. She licks them, she grooms them, she cuddles with them, all of that. The methyl groups went away. And then the kids had low anxiety. They became good groomers of their offspring. And they had low corticosterone levels, cortisol. So what this means is basically the way that mom treated the mouse when the mouse was growing up determined its methyl patterns. And then that determined how that mouse treated future generations. So maybe some of the behaviors or some of the things that seem innate or genetic in us might be epigenetic. So I might be partly the way that I am because of the way that my great-grandparents were. Maybe I'm very nurturing because my great-great-great-great-grandparents, I might go back that far, were nurturing, and now it kind of got passed down through the generations. All right, well, it gets a little bit more interesting than that even. This, by the way, is why we handle babies that are in like a neonatal intensive care unit, a NICU. So we still take babies out and rock them. Here it looks like this is a nurse. A lot of times nurses aren't able to do that. That's an expensive person for rocking a baby, but they'll just have volunteers. Sometimes there'll be people, and they might still, wear scrubs and whatnot. But they might come in and like, yeah, I'm grandma, I'm retired. I go and I rock the babies a few hours every week. So that's the idea behind that. This right here is another Science Daily article. It says, mother's touch may protect against drug cravings later. This is just kind of interesting. Okay, so the next thing we want to look at is this. Methyl patterns can be inherited. So this next part is kind of bizarre. So I'm assuming you were here for the day we talked about it in class, so it might not be too bad. But if it's the first time you're hearing it, then it is pretty bizarre. You might have to rewind and listen again. So this boy has Angelman Syndrome. So does this boy, this boy. They just happen to all be boys. Girls can have Angelman Syndrome also. And this boy has Prader-Willi. And so Angelman Syndrome is named after the first physician to describe the syndrome. Prader is, I think it's Andrea Prader and then like Heinrich Willi. So it's probably really pronounced like Heinrich, like Willi, kind of has that V sound. But I was born in Oklahoma and now I'm in Texas, so I'm calling it Willi. So Prader-Willi syndrome. Turns out they're both deletions from the exact same spot on a chromosome. So it looks like this. One thing that's puzzling about the way we name genes, on one hand it makes sense and on the other hand it's tricky. A lot of diseases are named based off of what happens if you don't have the gene or if you have a mutated version. So for example, I have genes in my body called eyeless, and I have the genes and they're not mutated. That's why I have eyes. But if those genes were mutated, then I would not have eyes. I would be eyeless. Picture a plant or something like that. They might have a gene called dwarf. And basically if you have good copies of the gene, then you're a typical Hythe crepe myrtle. But if you have a mutated version, then now you're a dwarf. So you're named off of not having it. So having a good Prader-Willi gene means that you don't have Prader-Willi. Lacking a good Prader-Willi gene means that you have Prader-Willi. So they didn't ask me to name this stuff. This is just how it is. So over here on chromosome, is it 19? Chromosome 15. On chromosome 15, I get a chromosome 15 from mom and a chromosome 15 from dad. So the red part up here is the Prader-Willi segment. And I should have a good Prader-Willi gene from dad, so I'm going to be fine. For mom, my Prader-Willi gene is methylated, so it's off. It's like it's not there. The only thing that's different is, in theory, if that methyl group were to leave, then I could turn it on. But otherwise, missing a gene or having a gene but it's off is kind of the same thing. You know, not having that file in my computer is about the same as having the file but not knowing where it's at to access it. So I get a chromosome 15 from mom and chromosome 15 from dad. Mom's chromosome 15 on the Prader-Willi part is silenced. On dad's, it's on, so I'm going to be okay. On mom's gene, I've got the Angelman gene. Technically, it's UBE3A, but I'm just going to call it the Angelman gene. So I should have a good copy from mom, and then my copy from dad is silenced. So here's where it gets weird. If I delete that part of dad's chromosome to where it's shorter, I deleted that chunk right there, then now that means I've got mom's chromosome, so here I am, here's the original parents, and then let's say I have a mutation. So now I've got a normal mom's chromosome, which means I've got one good Angelman, but I don't have a good Prader-Willi gene. So that means from dad, if the mutation came from dad, I'm going to have Prader-Willi syndrome. Over here, if I delete the same chunk of DNA, but instead of deleting it from dad's chromosome, I'm deleting it from mom's, then now I've got an off Angelman's, and I've got an on Prader-Willi. So now this kid is going to have Angelman syndrome. So again, here's the chromosomes. We delete a chunk. Well, we deleted a chunk of your chromosome 15. Are you going to have Angelman syndrome or Prader-Willi? Well, it depends. Did we delete a chunk from the chromosome you got from your mom or from your dad? And that determines which disease you're going to have. Well, what's the difference between these diseases? In Angelman syndrome, you typically are happy. Whenever you walk or run, you kind of have your legs and arms in front of you, Normally when people walk or run, like right now, if I was running, my arm goes behind me, my leg goes behind me. But if you ever look at like puppets, you know how they kind of, like these, like the puppets on the string? So you kind of have this sort of characteristic run, Angelman Syndrome. Low IQ, never speak, can still show emotion, you know, happiness or sadness or something, but very low IQ. Prader-Willi, on the other hand, is an insatiable appetite. You know, you have to lock up the kitchen. In extreme cases, maybe you have to lock up garbage because otherwise your kids will sneak down in the kitchen and like sneak food at night. So they kind of describe, you might remember that video that we saw in class, the boy kind of describes it as feeling like pins and needles in his stomach all the time. He basically is always feeling like he's starving. Two totally different diseases based off of where did the mutation come from. So those methyl patterns matter. Okay, now the next part. From here, we're going to skip this. This is a classic study. If you wanted to Google it, in Sweden, they had, I'm gonna try to pronounce this. If you Googled Epigenetics over Calix Sweden famine, you can find some information there. But I'm gonna go with a more straightforward experiment. In my estimation, a more straightforward experiment. We've got the Agouti Mouse. These guys are genetically identical. They are epigenetically different. So in this instance, what's happening is the mouse on the right has several genes, the Agouti genes, or the Agouti gene, that is methylated. So it's off. And that's why he has normal fur color, he's less likely to be diabetic, and he's average weight. This one over here, his Agouti gene, is not methylated. It's in the on position. And in this one, the on position causes disease. So here's kind of a picture. They're just showing, I've got the normal healthy mouse over here because he has some methyl groups attached to his DNA. This guy's obese because methyl groups are not attached to his DNA to turn it off. So they did an experiment. BPA, you've probably seen, like you might have some sort of water bottle or something like that and it claims it's BPA free. When I first got this, it probably had a sticker on it that said it was BPA free if it was a plastic one. And it turns out that whenever we got these mice, if we gave them BPA, then they became Agouti mice. So BPA seemed to be maybe either interfering with the ability to put methyl groups on or strip the methyl groups off, who knows. And so what this scientist did, Dr. Jirtle, if you wanted to Google it, it's Dr. Jirtle, Jirtle. These are all genetically identical. He gave them all BPA. When you give mice BPA, they are yellow and they're obese. But then he also changed their diet. So these ones on the end, he gave them folic acid, and then this looks like it'd be pronounced betaine or something like that, or betaine, but it's betaine. Yeah, betaine. And betaine has a whole bunch of methyl groups. Methyl groups are CH3. So again, when we treat everybody with BPA, the methyl groups are gone, and you look like the mouse on the left. But then, if we give everybody BPA, and then the mice on the right got something that could put in methyl groups like vitamin B12, folic acid, betaine, then they turn out normal. And what they found was, whenever you had these things in your diet, you were able to methylate those Agouti genes. But they're genetically the same. It also means you probably don't really want BPA in your plastics. In addition, we could go in and change how a mouse felt. We could, oh, that's actually a different experiment. So hang tight. Here's another example. Queen bees versus worker bees. Queen bees and worker bees are genetically identical. So there are genes that are on in worker bees. If the worker bee gets some royal jelly, then now that provides methyl groups that turn certain genes off, and now that worker bee becomes a queen, but they're genetically identical. Epigenetically, they're different. Genetically, they're identical. pretty interesting. This one back here was basically showing, I got these slightly out of order, that experiment where we had a mom that was licking the pups, like this. We were basically showing that we could take a pup that didn't have anxiety and inject it with medications that would add methyl groups, and we could turn it into an anxious rat. So adding and removing methyl groups really was what was causing the anxiety in mice, which is interesting because humans have anxiety too. And depending on where you read, we're about 85, maybe as much as 90% genetically identical to mice. So that's significant. Let's see. Transcription factors, promoters, activators, repressors. We kind of got into this before. I'll go over it again really briefly. So in order for a promoter to be on, let me check my time real quick. Okay. In order for a promoter to be on, not only does it need to be open instead of blocked, but also need to have like transcription factors land on it, TFs. And then nearby, there might be a regulatory sequence. That regulatory sequence might be an enhancer sequence, which binds activators. And that means the gene is going into the on position. Or it might be a silencer sequence, which binds repressor proteins, and that's sort of shifting the genes into the off position. It turns out to turn a gene on takes a whole lot of proteins. So here's a somewhat simplified version. So what scientists noticed was I might have a promoter and I might have a terminator, but then for a while we couldn't figure it out. There were regulatory sequences like way far away, like 1,000, maybe 10,000 base pairs away. And whenever it would or wouldn't bind a protein, would help turn the gene on or off. And so for a while, scientists were kind of perplexed. But it turns out, so here's my promoter, here's an enhancer way over here. When activators bind to it, my DNA can fold. And when it folds, now that regulatory sequence is near the promoter. So let's say this is my promoter right here. See if I can get this to stick at all. So my promoter is right here, the blue, and here's a regulatory sequence. Whenever I bind a protein, this purple protein on this regulatory sequence, my DNA folds, and now I make it more likely for RNA polymerase to land there. Except it's not as simple as just like one protein or two proteins or something like that. As you can kind of see in this picture, it's a lot of proteins. Here's another one. Right here is that purple thing is RNA polymerase, so that means this is my promoter. All these proteins are basically folding to encourage RNA polymerase to land there and turn the gene on. So between enhancer sequences and silencer sequences and all of that, and transcription factors, all these proteins are working together to basically say like, yes, this gene is on. The last thing that I wanted to mention about gene regulation in eukaryotes is that of microRNA and siRNA. So microRNAs are found, those are the ones that are found, let's see, yes, they are found naturally in cells. And microRNA works like this. So let's say I read this gene right here. This will seem a little bit weird at first. So I transcribe this, I unzip it, I read it, I edit it, and now that piece of RNA for this gene, I'll make it brown since that's brown. That piece of RNA is leaking out of the cell, leaking out of the nucleus, sorry. And so now, a ribosome can land on there and start transcribing. Man, translating, I was doing so great till the last like 2 minutes. So I transcribe a piece of RNA, it leaks out, I start translating it with a ribosome. Well, let's say that for whatever reason, my cell decides I don't want to translate it anymore, but it's already out there. It's almost like you sent the email, you wish you could take it back. So now I've got another segment over here, and it can make a piece of RNA that's fairly complementary to this. It won't be perfect, but if this was like, you know, ACC, then it would be UGG, and it'll basically bind to this, making it double-stranded RNA. Now my ribosome can't keep moving, so I've effectively turned the gene off. So there's multiple places you can turn genes off. You can turn them off at the DNA level before you ever do transcription. or after you've done transcription, you're in the translation phase, you can turn them off again. So this little extra segment right here is called a micro mRNA. siRNA do the same things, but they're coming from somewhere other than the cell, like maybe a virus. So I've got a quick little video clip of that, and then we'll call it done for this video. Let me find it real fast. I believe it's this one. Yes. So there won't be sound because I have my HDMI hooked up. So this is our DNA inside right here. We make a piece of RNA. This is my microRNA that I'm going to make. We make my piece of microRNA. And that microRNA is going to be complementary to, whatever other piece of RNA it regulates. So here's my regular piece of RNA. It codes for, make this bone cell grow. So that's what this is. That's the ribosome reading it. The microRNA can now land on there. Again, it's not a perfect fit. And I stop translation. So that's a way of turning genes off. And the reason this matters is we can now do this in a lab. So used to, if we wanted to do an experiment on mice and we wanted to see what it was like to, say, knock out the myostatin genes. You might remember the mighty mice that we saw in class. So this guy was big M, big M. This guy is little m, little m. Back in the day, you had to go through and turn off, or actually remove, you had to remove chunks of DNA. They called them knockout mice. Well, now what we can do is just go in and without changing their DNA, we can change them epigenetically. It's easier to change things epigenetically. And so we were able to make those mighty mice. In fact, over here, this follistatin is basically just a way of turning off the genes. So this guy is big M, big M. This guy is big M, little m. And this guy is little m for myostatin. And you can see the difference in muscle hypertrophy in those guys. You might remember we also had the whippets. The whippet on the left is little m, little m. The one on the right is big M, big M. So we can do this without actually changing the DNA. We just change the epigenome. So the overall idea of this unit is with epigenetics, we can acetylate histones that will tend to turn genes on. We can methylate DNA promoters that tends to turn genes off. And then the significance is we can pass that on to future generations. So it changes throughout your lifetime based off of experience, and you can pass it on to future generations, like we saw with the methyl groups with Angelman and the way that mom treated her pups getting passed on to future generations. So yes, you do just pass on your DNA to future generations, but maybe some of the stuff that you do in life still does get passed on as well. This is a myostatin Belgian blue cattle, so that's a little m for myostatin. All right, we'll call it a day with that video. Don't forget, 2009 number 4, 2019 number 7. Thank you.